Today, we have a distinguished speaker, very pleased to have Professor Pierre Baldi for this bioinformatics seminar. Professor Pierre Baldi is born in Rome, Italy, and then got a master's degree and the bachelor's degree of science from University of Paris in France, and then PhD in mathematics in California Institute of Technology, Caltech. Then currently he is a distinguished professor at the University of California, Irvine School of Information and Computer Sciences. He has many publications, over 200 publications, and he has several many awards, including the Fellows of Association for the Advancement of Artificial Intelligence and IEEE and ACM. He is also the 2010 Eduardo Caianiello Prize of Scientific Contribution to the field of neural networks. And he is also a fellow of ISCB, International Society of Computational Biology. So I have known Professor Pierre Baldi's work from when I was a grad student. Professor Baldi is really the pioneer of applying machine learning methods, especially neural network and deep learning, which is called nowadays, and from very early days. And I really remember that I see his eye-catching papers coming out on bioinformatics and other places. So he has a very strong influence in introducing machine learning, state-of-the-art machine learning into bioinformatics field. So I'm really pleased to have Professor Baldi here today. Professor Baldi will tell us about today deep learning in the biomedical sciences. Professor Baldi, please.
Thank you, Daisuke, and everyone else for having me. This is supposed to be very informal, so you can even interrupt me or put questions in the chat, and I will try to speak for about 40-50 minutes. Is that about right? And then maybe we can have some discussion, right? Okay, so deep learning, the biomedical sciences, it's a very broad topic. There are thousands of scientists working on this, so I will present some of the things I've done, but it's of course not comprehensive. And I certainly don't claim to have invented everything. But if you're interested in some of the things that I described, I recently published this book with Cambridge University Press, which has a lot of the theory we have developed for deep learning and the applications in the natural sciences, including, of course, biology and medicine, but also in physics and chemistry. All right, so the first, fundamental thing that one has to be aware of is that there is a two-way freeway between deep learning and the biomedical sciences. Of course, we think most of the time about the applications of deep learning to biology, to medicine, analysis of data at all kinds of scales, from the molecular all the way to the clinical. But of course, a lot of the ideas that we use in deep learning to develop deep learning methods come from biology, essentially from neuroscience, trying to understand the brains. I will, briefly review the history of convolutional neural networks, which are used for vision, biomedical imaging, but also other things. But, other recent trends like attention mechanisms are inspired from the brain, right? So it really goes both ways, and it has been very useful to me throughout my career to work in both directions and try to fertilize one areas with ideas from the other area. All right, so when deep learning start, I think one possible start for deep learning is shallow learning. And shallow learning is linear regression that was discovered. Least square method for linear regression were discovered by Gauss and Legendre at the end of the 1700s, essentially, right? And of course, they didn't have ideas about machine learning, about the brain. This was really about data. They were looking at astronomical data. But they came up with this idea: you have a bunch of dots, that's your data, and you're trying to fit a line or a linear space to the dots. And they discovered the mathematical equations that give you sort of the optimum, the best model in the least square sets. And you could argue that deep learning is just, linear regression on steroids to some extent, because you have data, except that now we have very, very large amounts of data in high dimensional spaces. We're trying to fit, instead of linear spaces, we're using very often nonlinear surfaces, complex surfaces, but the basic idea is the same. The two big differences are that you cannot solve for the best surface when you are in these highly nonlinear and high dimensional spaces. You need to do you have to do it by iterative optimization, so stochastic gradient descent by propagation. And the other thing that is very different is that you cannot really see what's going on. In the linear case, especially in 2D, you can see this, look at this with your eyes, and you have a feeling that you understand exactly what is going on, and it's explainable if you want. But when you are in, you're doing genomes, you are in a 3 billion dimensional space, and you have 7 billion genomes, you cannot really see things with your eye. And so things become very opaque and hard to interpret, and people worry about what is the neural network doing, etc. But that is not really that surprising. And if you think about it, by the way, when you are training linear regression, you're taking the information from the data and pushing it into the model, which is the two, in this case, the two coefficients that describe the line, the slope intercept and the y-intercept, right? Those are the synapses. You're collapsing all the information from the data in those two numbers. When you train a deep neural network, it's the same thing. You're collapsing all your data. You are sort of mixing it and collapsing it into every single synapse in the network. And from the synapses, from the value of the synapses, you cannot retrieve the data. It's an irreversible process. Even in linear regression, if I give you the red line, you cannot retrieve the blue dots, because the red line tells you only things about the average, the averages and the variance of the dot, the basic statistics. But it doesn't tell you anything about the precise location of the dots. So these are lossy methods for compressing information from the data to the synaptic weights. Deep learning itself, besides shallow learning, took off or began, if you want, in the late 40s, I would say, and was entirely inspired by neuroscience. So if you look at what we knew around 1940, 1950, we knew that the brain is the product of evolution. You have Darwin in the upper left. We knew already that the brain has specialized area, Broca, Charcot, etc. The brain has neurons, Ramon y Cajal, and so forth. This is Sherrington, who introduced the concept of synapse. We knew that neurons communicate with tiny electrical circuits. And on the theory side, you had information theory. Shannon had published his seminal work, and Turing had developed the concept of Turing machines and so forth, von Neumann architectures, Gödel. And finally, you have McCulloch and Pitts, the last two gentlemen on the lower row, who are credited with introducing a very simplified model for a neuron, which is this model here, the McCulloch and Pitt neuronal model, which is what we use in deep learning, essentially, where a neuron is a simple device that computes by taking the weighted average of its inputs and then transforming it through an activation or a transfer function into an output. And of course, we have many different activation functions that we use. If you use the identity, you have a linear neuron. Otherwise, you can use a sigmoidal function f, like a logistic or a tanh. You can have a threshold function, sin h, or a heaviside. And now you have piecewise linear functions like ReLU and so forth, right? So we have a whole zoo of activation function that we use, but that's the basic model that is the basic building block of neural networks and deep learning. And again, the emphasis, I described how the input-output is processed in this machine, but the key thing to realize is that there is this set of weights, the synaptic weights, which is where you store information. So you have both storage and computing that are, intertwined together. And this is one of the reasons why biology is so efficient energetically, by the way, 4 orders of magnitude more efficient than our computers. Processing and storage are completely intertwined. Again, the storage is very opaque. It's very different from the Turing tape model, where you have a physical substrate and you're putting information precisely at discrete locations, like even DNA is a Turing tape. This is very different. You take your information and you shatter it, and you put little bits of it in each one of these weights in a very opaque manner. So don't be surprised if these little machines are hard to understand. It's their nature. It's actually their nature. It's a very different way of storing things. So 1950, you could start asking, where do we store information? In the brain. Think about your telephone number, the first digit of your telephone number. Where is it in your brain? The truth is that nobody knows, even today. But the idea came, and even Ramon y Cajal already had thought about that information, one place information may be stored is in synapses. It may not be the only place. Today we know about epigenetics, so it could be that the information gets stored also in the DNA ultimately through epigenetics. But for fast response, it has to be in the synapses, because that's where you can influence the activity of multiple neurons, right? If you have to go in the DNA and take out the information from the DNA, you wouldn't be able to recall your telephone number in less than a second, right? So synapses is a good guess for storing information. But the question is, how do you do it, right? That's the mystery. If you think about it, synapses are very small. So to really impress you of the fundamental problem of learning here, I'm going to rescale them by a factor of 1,000,000. So multiply everything by 10 to the 6. You take a synapse, it's 10 to the minus 7 meter, you multiply by 10 to the 6, it becomes 10 to the minus 1 meter, that's 10 centimeters. It's the size of your fist. So imagine you are a synapse. Imagine your fist is a synapse, and you're trying, you're at Purdue University, and you're trying to learn how to play tennis or how to play the violin, and ask yourself, where is the tennis racket? You rescale by 10 to the 6, maybe the tennis racket is in New York, okay? So imagine the problem of this little fist in your office that has to decide whether to make itself stronger or weaker so that you play better tennis, when it doesn't know about tennis, it doesn't know anything about mechanics, Newton laws, nothing. It doesn't see the racket is in New York, and it doesn't have a Google where it can ask, what should I do, right? So that's deep learning. That's the fundamental problem of deep learning. You have this blind synapse, completely stupid, completely unaware of what the organism is trying to do, and yet it has to take a decision, decide whether strengthen, weaken itself, or whatever, change its biochemical parameters, so that at the global level, at the behavioral level, your behavior is more intelligent, better adapted, and so on and so forth, right? So that's the center of the deep learning problem for biology and also for neural networks. One of the first person who thought about this fundamental problem is Hebb. He came up with the idea of Hebb rule, basically the idea that neurons that fire together wire together. It's a nice rule because it's local, right? You have a synapse, it doesn't know about the tennis racket, doesn't know about New York, but probably it sees its immediate neighbors, the pre and the postsynaptic neurons. So let's look at the correlation and let's decide whether to go up or down based on that correlation, right? So it makes a lot of sense for being a local rule, completely blind, completely stupid. But the problem is, suppose you do this and you have 1 trillion synapses and you apply this stupid rule, why would you get nice, organized, intelligent behavior, right? Nobody has been able to take any kind of network, apply this rule, and get it to work on any kind of complex task, right? So A few years later, Rosenblatt came up with the Perceptron algorithm. So it's the training algorithm for a single neuron, maculocampid neuron. If you study the perceptron carefully, you will see its gradient descent. He basically discovered gradient descent for a single neuron. And then next big milestone is Hubel and Wiesel, their work on the cat visual cortex, discovering that there are neurons in the primary visual areas in V1 that behave like edge detectors, and they can detect edges at all possible orientations and all possible locations in the visual fields. So it's the idea of convolution, convolving the input with these arrays of filters. There is a very nice little YouTube video. I'm not going to play it, but if you have never seen it, I recommend you go on YouTube and you look at it. You will see their video from their experiment where they are moving these bars at different orientations in the visual field, and they are recording the sound of the neurons through a microphone. So you hear the neuron firing when the bar is at the right location, right orientation, and when it's at a different orientation or different location, the neuron is completely silent. If you've never seen this, it's very striking. I highly recommend it. A few years later, Fukushima in Japan took the idea of Hubel and Wiesel and came up with the idea of what we call today convolutional neural networks. Basically, you use the maculocampid neurons, you build an architecture, but you import the discovery of Hubel and Wiesel by saying that you're going to have, in the early stages of processing, you're going to have arrays of neurons that are acting like edge detectors at all possible locations or possible orientations, and so on and so forth. And Fukushima had the idea of learning the connection of this network, but he didn't know what to do. So he went back to Hebb and he said, well, let's learn the connection using the Hebb algorithm, the correlation, right? And nobody has ever been able to get this to work. Today, we understand that you can prove mathematically that if you apply Hebb, you're trying to learn the MNIST data set or ImageNet with a convolution CNN, and you apply Hebb rule to all the layer, you will never be able to succeed. We have a paper proving this mathematically. So to learn again, you need something else. And the something else is just gradient descent, stochastic gradient descent. It's called backpropagation for neural network because of the chain rule, you just have to go from the output layer backwards towards the input layer and change the weights. It was discovered, rediscovered, co-discovered by a number of people. I'm not going to give credits to anyone here, but the idea is very simple. Newton could have done it had the problem been explained to him. It's gradient descent and it's the chain rule. And ultimately, when you have to adjust the weight, the rule looks a little bit like Hebb. It's a product, basically, there is a learning rate times the product of two quantities. One is the activity of the presynaptic neuron, OJ, here. And one is, if it was Hebb, it would have to be the activity of the postsynaptic neuron, so OI. But no, that doesn't work. What you need is the postsynaptic backpropagated error, which is a signal that comes from the top layer all the way to that neuron. Okay? Once you have this, things start to work beautifully. You're probably familiar with Yann Lequin's work on recognizing digit. He became famous, in particular, for making this data set public, which was very useful for the machine learning community. It was widely used to test all kinds of algorithms. By today's standard, it's way too small, so we have other data sets. But in the 90s, early 2000s, this was very useful. But other people were doing these kinds of things. And I think modestly, we had the first application of the convolutional neural network to biological images. This was a fingerprint problem that we tackled in the late 80s, early 90s, using a convolutional neural network, where you put, today you would call it a Siamese convolutional neural network, where you put two images in the input, two images of two fingerprints, and it decides yes or no, this is the same person, right? The same finger from the same person. So that was, late 80s, 90s, and so forth. And today, basically, this is what is working on biomedical images. I'll show you example of things we're doing now these days, but with much bigger networks, of course, ideally much bigger data sets and of course GPUs, which have played a very big role in speeding up things, and also the software. So you have very well-maintained, robust library like Keras, TensorFlow, PyTorch. You're probably very familiar with those, maintained by Google, Facebook, etc., which everybody uses and makes things very, much easier for developing applications. Okay, so today we have big data. I'm gonna skip all this. These are DNA sequencer, for example, one type of instrument. I'm sure you have probably some on campus where you are. They produce terabytes of data. Once you align the reads, you get entire genomes that looks like this. And of course, this is not readable by humans. So you need, again, computers, deep learning to understand this kind of data. Same thing for imaging and so on and so forth. But basically, you can apply deep learning because it is like linear regression. You can apply linear regression to anything, economics, psychology, physics, anything. You can do linear regression if you have the data, right? Same thing for deep learning. So it's not surprising that you can apply it in the biomedical space, in bioinformatics, at all levels, all scales, whether you work on small molecule drug discovery or on genomic data or gene expression data, metabolomic, any kind of omic data, epigenetics, all the way to systems biology, electronic medical record, clinical data, population data, all of those. If you have the data, you can try to apply deep learning to those problems. So it's a very rich field, and again, as I said, thousands of people are working on these things today. One point that I want to stress, though, is that CNN are good for images, and more generally, feed-forward networks are good for problems where you have data of variables of a fixed size, like images, or your images have the same size. However, there are many problems, especially in bioinformatics, where you get data that has variable size. Sometimes you have a protein may have length 30, sometimes it has length a few thousands, right? So variable length in the sequences, variable size of trees, if you're doing phylogenetic trees or other types of trees. And also they have structures, right? Phylogenetic tree, there is a tree, the sequence has a sequence structure. You're dealing with molecules and drug discoveries, you get labeled graphs, small labeled graphs, but of variable size and so forth. So how do you handle those with deep learning? You cannot do, in general, it's not a good idea to use feed-forward network on those objects because they're variable size. And then how do you input the size into the deep learning? So we have developed method for doing that. We call them recursive neural networks. It's a little bit different from recurrent networks, so there is a subtle there. But today, a lot of these methods are also rebranded as graph neural networks. But I'm going to give you the basic idea here. And there are two techniques for doing these kinds of networks. One that I call the inner approach and one that I call the outer approach. And I'm going to show you on simple example how they work, and you will understand what I mean. In the inner approach, you need to have a DAG, a directed acyclic graph, and you're going to crawl the graph using your neural networks inside the graph. In the outer approach, you build the graph that is sort of orthogonal to the data, and instead of crawling the graph of the data inside, you're growing your neural networks in a direction that is orthogonal or outer to your data. This may sound a little bit esoteric. Let me give you an example. So suppose you're dealing with sequences. A standard model for sequence, probabilistic model, is an HMM, right? So you write those little DAGs. These are also called Bayesian networks or graphical models, but basically a acyclic graph, where the idea here is you have a machine that moves from a state to the next, and when it's in a given state at time t, it produces a symbol or a letter from a certain alphabet, right? That's the basic structure of an HMM, the hidden Markov model, or even when you do Kalman filters, you have this kind of architectures, right? But the point is that you have a directed acyclic graph. And so if you want to refactor this in terms of neural networks and deep learning, what you do is you put neural networks on the edges. So I put neural networks on the edges. That means that the state at time t becomes a function of the state at time t minus 1. And this function is computed by a neural network. So here you have a vector that represents the state at time t. and it goes through a neural network and it produces the state at time t+1. Okay, and then you have another neural network that goes from time t+1 to t+2. Of course, if you want to shrink the number of parameters, you may reuse the same neural network. You may do what is called weight sharing, and so you can have a single neural network that takes care of the propagation of the hidden states. Likewise, when you are in a certain state at time t, you have a second neural network that tells you what is that you produce at time t when you are in that state. And again, you can use the same neural network at different times t for producing the symbol, right? And so basically you have this HMM. Now I've converted it to two neural networks, two basic neural networks, one for the hidden transitions and one for the emissions of the system, right? And if you draw this, you will see that you get a big neural network when you unfold everything in time. It's basically, because the graph is acyclic, it's just a gigantic neural network that you can backpropagate information through, so you can train it by backpropagation. Okay, so that's the idea of the inner approach. What about and you can do this, of course, for more input-output HMMs, factorial HMMs, bidirectional HMMs. So if you know about graphical models, you can take all those very nice probabilistic models and convert them to neural networks. In the outer approach, remember there's a second approach. The outer approach is slightly different. Imagine that you have your sequence at the bottom here, this sequence of blue dot. And now you're going to grow, instead of using neural networks inside the sequence, you're going to grow your neural networks in a direction that is outside, perpendicular to the sequence. And so you're going to have all these blue neural networks, these blue triangles. Each one is a neural network that looks at a window of inputs in the sequence, maybe the first three letters or the first three words or whatever you're looking at. And then you have a second neural network that looks at the next set of three words and so forth. And you can do weight sharing. This is why all of them are blue. It's the identical weights inside each network. You're applying them at each position. So basically it's a convolution again. You're applying the same position, the same operation at each position in the sequence. And the output of the blue networks are certain vectors. And now you can do the same thing on those vectors using the green neural networks. And you can stack many levels. You can have a red level and so forth. The idea being that if I look at the output of a red neural network, it is influenced by a bigger window in the original input sequence than the blue neural network. So as you go up in the stack, the neural networks are able to see more and more of the context of the long-range interactions that may be present in the sequence. Okay, and on top you get again your prediction, whatever it is you're trying to predict. It could be protein secondary structure, it could be, you know, other things. You can backpropagate. There are no directed cycles, so you can backpropagate information and train these architectures. All right, so for many years we came up with this idea of recursive inner and outer methods by working on the problem of protein structure prediction, which of course has been in the news with AlphaFold and using deep learning methods for this problem. But the basic ideas behind AlphaFold are not really new, and they've been developed on this problem. So if you want to predict protein structures, one idea using deep learning, the fundamental problem is that the structure of the protein does not depend on rotation and translation, right? So if you move a protein around, the structure remains the same, but the XYZ coordinates are changing, right? So if you think of taking a neural network, giving it the input sequence, the primary sequence, and predicting XYZ coordinates, that's not going to work very well because of this reason. The XYZ coordinates are not canonical. They depend on the frame. So you have to find a way of making a canonical, translation rotation invariant representation of the problem. And so the way we did that for many years was to start from the primary sequence, predict many properties like secondary structure, relative solvent accessibility, and so forth. And then from there, predict the contact map or the distance map, which is a translation rotation invariant representation of the protein. If you don't know what a contact map is, for instance, imagine It's a matrix. Imagine putting the sequence on the x-axis and on the y-axis, and then in position ij, you put a 1 if amino acid I and amino acid J are close to each other in the 3D structure. Otherwise, you put a 0. So you get a very sparse matrix. It has 1s on the diagonal because every amino acid is covalently bonded to its neighbor. But then it's symmetric, and it's very sparse because, you know, most amino acids are not in contact with each other, but you have a few long-range connections that are represented by the dots in this corner of the contact map, which are the ones that are important for the structure and difficult to predict. These are amino acids that are far away in the sequence, in the linear sequence, but end up being close to each other in the 3D structure when you downloaded it from the PDB, right? From X-ray diffraction experiments. And then from the contact map, the distance map is the same thing, but instead of 0 and 1, you put the actual distance between the corresponding amino acids, right? And then from this, you can try to predict the 3D structure. And so there are interesting questions in different stages of this algorithm, but the most difficult part is to predict the contact map or the distance map. That's really the most the key step in this pipeline, and this is also what is behind AlphaFold. So you can apply deep learning to predict the secondary structure, for instance, or the solvent accessibility, to predict the contact map, and so forth. So for predicting secondary structure, for instance, we developed these methods here. What you have in the drawing above, it's what is called a bidirectional input-output HMM. It's just a slightly more complicated version of what I showed you before, where you have both inputs and outputs, right? UT is an input at position T, YT is the output at position T, but then you have hidden states in the forward direction, FT, and in the backward direction, BT. You can easily check that this graph is acyclic. So now we are going to replace it with neural networks. And if you do a little calculation, you will see that you need 3 neural networks in this case. One neural network that computes YT as a function of BT, FT, and UT. That's the production neural network. And then you have one neural network that does the backward propagation. So it predicts BT as a function of BT plus 1 and the input, UT. And then you have the forward hidden chain, where you have a neural network that predicts FT as a function of FT minus 1 and the input UT. And these are all shared at all position T's. So you basically have 3 neural networks. If you expand it in time, you would get this sort of architecture, which is very big, right? Each triangle, dark blue, light blue, all these are neural networks themselves with many layers and thousands or millions of parameters, but they are basically being stacked to process the data at each time T, right? So it's a cyclic, you can train that by back propagation and so forth. So that's what we developed in the late 90s. We've kept refining it. And if you look at secondary structure prediction, for instance, you can see that it started in the late 70s with Chu and Fassman, who came up with very simple statistical rule, based on how often a given amino acid, how often alanine is an alpha helix versus in a beta strand versus in a coil. Same thing for all the other 20 amino acids, right? So they were the first to come up with a statistical rule. It was about 60% correct. Kian and Sinowski were the first to use deep learning. They used neural networks in the late 80s for secondary structure prediction. Prediction accuracy went up to 64. Roast and Sander did a nice piece of work in 94. They introduced profiles and so forth. 99, that's where we introduced these recurrent networks to do this. Was 78%. Anyway, today we are about, between 95 and 100% correct using these techniques and other things that I don't have time to describe. So the problem is basically solved because you cannot get 100% correct. There are errors in the PDB. There is proteins that do not fold spontaneously. They need chaperones, etc. Or some proteins need additional things like metal ions to fold properly, which is not part of the input, and so forth, right? So secondary structure was solved, has been solved. And now, of course, prediction of contact map, you can again use the same ideas of inner approaches. So now instead of having these 1D sort of models like HMMs or bidirectional HMM, you have to do the same thing in 2 dimensions. You think of the contact map as a 2-dimensional object. So now you are going to have an input plane, an output plane, and you have 4 hidden planes for propagating information in all along towards the 4 cardinal corners, basically northeast, northwest, southwest, and southeast. So it's exactly the same idea as a secondary structure, but I'm doing it now in 2 dimensions. It's generalization in 2 dimensions. When you look at one vertical column in that object, you get this picture where the output at position ij, 2-dimensional position ij, is a function of 4 hidden vectors in the 4 hidden plane below it and of the input, right? And then let's say you are in the northeast plane, northeast lattice, the value of the northeast vector at position ij depend on its 2 immediate neighbors in the lattice and on the input. So again, this becomes 5 neural networks when you unfold it. And again, you can check everything is acyclic. You have here the equation, the output at position ij is a function of the input ij, the hidden vectors in the 4 hidden plane, and you just parameterize this function by a neural network called NO, N for output, O for output. Same thing for the 4 hidden plane, and you get a method for predicting things in 2 dimensions for contact map or distance map. If you want to do the outer approach for contact map, you can do that. Oops, I'm not able to change. Oh, there it is. You can do the same thing. So the outer approach would mean that you take your contact map, your plane, and then you are stacking neural networks on top of the plane to try to predict distances or contacts. And you can do this with many levels and so forth. So that's the outer approach. That's also something we developed and published in 2012. That's the method that DeepMind has been using in AlphaFold with many variation and interesting ideas. But that's the basic idea. You stack neural networks over a 2-dimensional sheet in order to predict distance and contacts. All right. So then there are many applications beyond protein structure prediction. You can apply deep learning methods to molecular data, chemical data. So if you're doing drug discovery, you want to predict whether a given molecule has interesting properties, whether it's soluble, whether it's toxic, whether it binds to a particular protein target, and so forth. The interesting thing about small molecule is that they have very many different ways of representing them. You're probably familiar with the labeled graph representation, which is in the upper left. The problem there is that the graphs are variable in size and they have labels, so you have to use recursive method, inner or outer. You can also represent small molecule using strings. If you're familiar with that representation, these are smile strings. So all the technique, again, these are variable length, but all the techniques, deep learning techniques for strings, if any of you works on strings, on language, we have now things called transformers. You can apply transformers to smiles, strings. You can also, there is a way of representing molecule using long binary vector, very sparse, called fingerprints, where basically, you put a 1 if and only if a particular configuration, a particular functional group is present in the molecule or not, right? And very often you use a fairly large basis. So these vectors can be, thousands, if not hundreds of thousands of component in length, very sparse. Most of the time you have 0, but here and there you have 1. So you could use this representation for applying deep learning, or you can look at molecule as what it is, which is a 3D object. And so you can represent it as a list, actually as a set of XYZ coordinates for the center of the atoms, right, in the molecule. That's another way to describe it. And so you can apply inner and outer model and feed-forward networks to any of those representation. And if you can find the data, train them on solubility, toxicity, whatever you want. So this is also an area where there is plenty of work that can be done We have published paper on that. I'm not going to spend more time. But that's the inner. This is the description how you do inner approach on molecules. It's a little bit challenging because the graph is not acyclic, right? If you have a benzene ring, you get a cycle. There is no obvious orientation, so you have to do a little bit of work to address that, but it can be done. Or you can use the outer approach on molecule that also has been done. You can apply the similar techniques to prediction of chemical reactions. I'm not going to spend time on that, but that's another area where you can apply deep learning on chemical reaction. This is an example of work from some of my colleagues, Xiaoyi Xie and his collaborator, applying both convolutional neural network in the first stage of processing, but then also bidirectional ideas like the one I showed you using something called LSTMs. But this is applied to DNA sequence and trying to predict tissue-specific chromatin marks in the DNA sequence. You can try to predict, binding site for certain transcription factors or whatever, promoters, anything that interests you in the genome. You could try to apply these techniques at the genomic level. There are, of course, many other applications in the genomic space. We are using them to study circadian rhythms, right? So if you do omic experiment in a circadian way, every 2 hours, gene expression in mouse or any other organisms, you collect data every 2 hours, then you have the expression levels of, say, 20,000 genes collected every 2 hours. You want to know which genes are being expressed in a circadian fashion, right? Which genes are oscillating with a 24-hour period. So we are applying deep learning to that problem, for example. But to finish my talk, I want to move to the biomedical imaging problem, where you can apply those CNN of Fukushima, but with backpropagation that I told you at the beginning of my talk. And that's really an area that basically is very mature now. Almost any problem in biomedical imaging, people are developing deep learning methods, convolutional neural network that perform at least as well as humans, and very often a little bit better, actually. So first example, this is a collaboration with scientists at UCI who have developed a system where you have microfluidic chambers where you can grow blood vessels. So what you see here in green are the blood vessels growing in these little microchambers. And you can also put cancer cells. The blue here, what you see in blue are the cancer cells. You can put a drug, and then of course you want to know, did the drug change kill the cells or reduce the cells, or the opposite? Did it help the cell to proliferate faster? And did it change the vasculature? All these things, you don't want to have humans, this is high throughput technology, you don't want to have humans looking at these images and making all those decisions. So you can train neural networks to analyze these images very rapidly and tell you, for instance, whether the number of cancer cell has increased or not, right? So that's one example. I'll skip this one. Another example, this is from not microscopy, now we're with X-rays. These are mammograms. These are X-rays of the breast, which are performed routinely by the millions in the US and worldwide to detect breast cancer. So you can train the networks to predict whether there is cancer or not, or other things. But what we're doing here is something completely different. We're trying to predict the level of calcification. So it's really a regression problem. We're trying to predict the level of calcification in the blood vessels in the breast, because a high level of calcification is potentially a sign for heart disease. So as a byproduct of getting a mammogram, we could detect, if you have a very high level of calcification, it would be an additional screening tool, and we would be able to warn the patient that maybe he or she should undergo additional tests for heart disease besides being screened for cancer, right? This is an example where detecting cancer metastasis in the spine. I'm not going to spend this time on this. These are examples on gliomas in the brain. What I want to show you now is applications to videos, because that's also very, very interesting. So you're all familiar with colonoscopies. where doctors go in the colon and they take videos of the colon, and they're looking for polyps, which is what you see in the green box there, which this is where colon cancer develops, right? So typically during a colonoscopy, a doctor will remove all your polyps, and then they will be also sent to pathology to see if any of them was malignant, right? But as a precaution, they are removed. The problem is that the surface of the colon is roughly the surface of a tennis court, and the colonoscopy is done in 5 minutes. So it's not uncommon for doctors, even experts, to miss polyps, sorry. So it would be nice to have an AI method, a deep learning method that can detect polyps automatically. And we did this in collaboration with doctors at UCI, where they gave us images of the colon, some containing polyps and some not containing polyps. So we trained CNNs on predicting yes or no, there is a polyp in the image. We can also put bounding boxes. There are techniques for doing that. If you're interested, I can tell you more about that. But the point is you train it on images, and then you ask, how can you do this, use this in video, right? Well, the point is that when you do the training, that's very slow. It may take a week, 2 weeks, depends on your hardware, it depends on how much data you have and so forth. But once a neural network is trained, to process an image is very fast. It's something like 20 milliseconds. So if you give me a video, I just take, 10 frames, 15 frames per second. That's plenty to do prediction 15 times per second of whether there is a polyps or not, right? You can even use evidence from neighboring frames to make more robust inferences. So I'm going to show you if I can do that. Oops, doesn't seem to. Oh, it looks like I cannot show you movies. Is there some? Oh, here it is. So what you're going to see is a collage of colonoscopies. I know it's lunchtime, so don't be too worried. It's not so bad. In Purdue, it's lunchtime, but you see the AI, which basically AI means just a neural network, right? A convolutional neural network putting a bounding box in real time around the polyps in colonoscopy. And this is actually being used now in clinic. It's being deployed. And it is essentially as accurate as human expert, possibly a little bit better. And, as you give them more data in time, they have a chance for getting, slightly better, although they are, in the high 90s correct accuracy already. Another example now, it's in eye surgery. So in the eye, you have a structure called the TM, which is called the trabecular meshwork, which is actually quite difficult to see. It's a band, you can see it in the lower left. It's quite difficult to see, and it's a very important landmark for eye surgeries. So you would like, again, in real time, to be able to delineate the position of the TM. And I'm going, if I can do it, show you a movie. Yeah, here you see in real time our system. Again, it's essentially a convolutional neural network, which is delineating the TM, helping the doctor find a TM during surgery. This is not deployed yet. It's still at the prototype stage. How much time do I have? Well, I think, just the last one to show you also what you can do. Of course, you can go into veterinary medicine and apply the same ideas to radiographic or microscopy images or whatever, video images of animals. And we have worked on a disease which is called the valley fever, which is becoming more and more important due to global warming. It's caused by a fungus that can be inhaled from dust. Typically, it happens with dogs. They can get this disease, but also other animals. They can even pass it to humans. So we work with a company that is interested in dogs. And again, you can get images of the lungs of dogs, and you can build the convolutional neural network that says yes or no, valley fever, this dog has valley fever or not, and can even tell you, what is it in the image that seems to be important for making the decision of the presence of the disease. So it can even help doctor to further, zoom in into particular areas of those images and assess the diagnostic. Okay, I think I'm out of time. I'm going to stop here, and perhaps we have some questions or a discussion or.
Okay. So, all right. Thank you very much, Professor Baldi.
You're very welcome.
So, well, from audience, any questions?
There's something in the chat. Let me see. What information you get from using backpropagation unit, and does it have a relation to time? So when you do backpropagation, the information that you send back to the synapses is an information about essentially about the performance of the network. How well is it doing? What is the error, right? So when you train the neural network, you have examples, you put them in, you get an output, you compare them to the true value. You need to have labels in supervised learning at least. And then you have some error, right? It could be the quadratic error like Gauss and Legendre. They use the quadratic least square error, right? The L2 norm. That's called the least square error. And so that's an example of error that we use. There are many others, but basically you get an error and you want to do gradient descent, you want to minimize that error. So when you do that calculation, how do you compute the derivative of the error with respect to the weights, the synaptic weights? That's the information that you have to send backwards in the network, right? Does it have a relation to time? No. If you're looking at an image, there is no relation between time and the backpropagated error. It's really the chain rule applied to a neural network. So because of the chain rule, you have to do it first at the top of the network and then go to the layer right below it and so forth. And little by little, you go down the network in the direction opposite to the direction you use when you send things, information through the network in feedforward mode. Other questions?
I have questions about basically all your medical work. So I also had some collaboration with biology people who give me some images. But one of the issues I also always encounter is the amount of data they can provide. So I always need to ask them, okay, I need a little bit more data. 50 is well, doable, but not enough. Can you give me more? So there's always, actually, I need to ask them to work a little bit more to give me more data before I can do something. So in your case, do you use more machine learning techniques to augment it or do something? Or how do you deal with those data problems?
So that's a very good question. That's indeed one of the main problems. The data is really precious, and getting the data is really sometimes a bottleneck. So when you don't have a lot of data, you do data augmentation. We do it all. Even if we have a lot of data, we'll still do data augmentation, which means we create additional data by perturbing the data that is given to us. Sometimes you do it with rotations, with flips. You had Gaussian noise. That depends on the problem. How do you augment the data? That's one direction. But the other direction that I think has completely changed this field is what is called pre-training or neural network surgery, or whatever you call it, you want to call it, which is that today you have neural networks that have been trained on, general purpose computer vision problem. And these networks are very large. They can have, let's say, 1 billion parameters. They have been trained to recognize cars and trees and pedestrians, whatever. But they are publicly available. So you can download those networks, and all the lower layers, all the filters are still very good because they are general purpose vision, right? Even a doctor, radiologist that looks at an image, all the basic machinery that is using in the brain is this general purpose visual system that was trained to recognize toys and people and other faces and other things. So you take these big architectures that have trained on completely different problems, but you can still use them. What you have to do is change the top. So you retrain at the top using the 50 or 100 images that the doctor gave you, but you keep all the rest from already trained, and that works very well. So you just retrain the top for the specific task, whether it's cancer or whatever you're looking at, and that's a very useful technique that is widely used today. And then there is the problem of how do you convince doctors, hospitals to create more data, to label more data and share more data. You know, that's an interesting problem for computer scientists in terms of building infrastructures, and also from the ethical, legal, societal point of view, how do you address those problems? And some people, it's not at all what I do, but some people are working on that. You know, within the University of California system where we are, you know, there is 10 large medical centers, and it's very difficult to share data between medical centers. You know, people are working on it. They always tell me, oh, it's almost ready, or it's ready, it's happening, but I haven't seen it working smoothly yet, right? It's already very hard to get data from my own medical center at UCI, let alone from combining data from UCLA and UCSF and UC San Diego and so forth. So it's a challenge, but of course that's an area that is progressing, and definitely I think the technology is ahead of the reality. I mean, you really could have massive deployment of convolutional neural network in biomedical imaging. It is happening, but it's still a relatively slow process. But it will happen, of course.
Okay, thank you very much. Any other questions?
Majid.
Yeah, Majid, sure.
Yeah, thank you very much for such an amazing body of work. It's really fantastic. So I have a little bit of a naive question. So when things are not like images and you went through them like sequences and drugs, structures, where they don't have a great way of representing them, in your experience, what are the effect of representation on generally the ability of these deep neural networks or deep models to accurately define or predict the things that you're looking for? Do you think that the representation is a major piece of learning, or having a good representation is a major piece of learning, or they could also learn the representation?
Well, I think it's both. I think the representation is very important. And what we are discovering more and more is that deep learning methods can learn the right representations. So those two things go together. But it implies that you need, fairly complex or fairly big comprehensive neural networks. So up to recently, you were only training the part that went from the representation to the decision to the prediction, whatever, right? And that works well only if you have the good representation. If you have a bad representation, you're going to struggle, right? So now if you want to learn the good representation, you have to put another neural network, essentially. You have to make your neural network bigger so that it can first learn the representation and then do whatever task, prediction, classification you're interested in. So I think you can do everything with deep learning, but you need, good sized deep learning networks, you need GPUs and so forth.
Thank you.
Any other question? I have one more question, more closely related to proteins. So now the structural protein seems to be fairly well predicted, but now, maybe in biophysics, people started to work on cell level molecular dynamic simulations of motion of molecules in a large cell. Are those kind of like a more physical motion of many proteins, or a drug molecule in some system, in some larger system, can be also handled by predicted by deep learning?
Yes, so that's another area, you know, where a lot of work is going on, which is using deep learning methods to speed up or approximate slow but detailed physical models. People are doing this at the level of Schrödinger equations, you know, DFT, speeding up DFT using deep learning. People are doing this at the climate level, you know, Navier-Stokes equation, essentially, climate, weather prediction. They're putting neural networks in there. So it has molecular dynamics. So it happens at all scales where people are taking these complex physical processes where maybe we know perfectly or almost perfectly the microscopic law, but when you're trying to compute what happens, it's very slow, even today, right? It's very difficult to predict chemical reactions using Schrödinger equations, even today, especially if you want to do it in high throughput. So people are just, you know, using the physics to build the training set, and then basically using deep learning to approximate the training set very fast. So it's a whole little industry. A lot of people are working on that. And again, at all scales, from Schrödinger equations all the way to galaxies and the cosmos, you can apply these methods because, again, they are generic. It's like linear regression, right? It doesn't matter if the data comes from astronomy or from, you know, quantum mechanics.
Okay.
But it's a very good point.
Yeah. Any questions from audience? Maybe some of from my lab. I see people from my lab also. It's a very good opportunity to communicate with Professor Baldi.
Can I have a question?
Okay.
Oh,
Yes.
thank you for your great presentation, Professor Baldi.
You're welcome.
I've heard that applying machine learning and deep learning into the sequencing expression data, like RNA sequencing data, we usually face recursive dimensionality because, you know, there are like thousands of cells and thousands of genes in the data. So I want to ask you if there was an experience for you to apply machine learning or deep learning techniques in the expression data directly, and or how did you solve the recursive dimensionality problem?
Again, it all depends exactly on what problem you're looking at and what data you're looking at. We're looking, for instance, at circadian rhythms, right? So we look at gene expression and we want to determine whether a given gene is being expressed in a periodic manner in a given, you know, organism under certain conditions, right? So you may have 20,000 genes. It depends how many time points you have, how frequently the genes were sampled, and then it depends on the replication level, you know, how many mice were killed at each time point, and it depends on how many such experiments do you have, right? How many different conditions. So you could go from a situation where you have a little data to a situation where you have plenty of data and everything in between. The interesting thing we discovered in that project, but this is just specific to that project, is that we trained the system using artificial data. This is the only example I know where we can train a neural network on completely artificial data, because what we do is we take, let's say, a sine function, which is periodic, and then we sample it. Maybe we add noise, and that's an example of a periodic time series. And then we take something that is like a line that keeps growing straight, we sample it, we add noise, we do pseudo replicates, and that's another training example. So we can generate a lot of data like this, periodic, non-periodic, very noisy, less noisy, and then train a deep learning system to predict whether it's periodic, what is the period, what's the amplitude, all those things. So we have a system that is completely trained on synthetic data. We test it on biological data, of course, but it's trained on synthetic data, and so we can have as much data as we want. Since we are making the data, it's not we're not data limited for that specific problem. but even very basic question like differential analysis, is this gene overexpressed or underexpressed between these two conditions? That is a typical problem where you have 20,000 hypotheses in the mouse genome, and you do have a multiple hypothesis testing problem. This comes up all the time, right?
Thank you for your answer.
You're welcome.
Okay, thank you very much. Any quick question? If not, let's thank Professor Baldi again. This was a really great opportunity for us to learn a whole perspective of deep learning and the recent application of biomedical and science problems. Thank you very much.
Thank you very much, Daisuke. Thank you, everyone. Email me if you have any questions. Bye.
Thank you very much.
Bye.
Bye.